my pleasure to introduce our speaker today, who's Jason Hartford from computer science. So he is Dame Kathleen Ullenshaw, I think I've pronounced it correctly, fellow in Center for AI Fundamentals, Department of Computer Science at Manchester. And also he is a research unit lead at Valence Labs. And I guess he's very happily managing academic and industry roles. So Jason has a PhD in machine learning methods for causal inference from University of British Columbia. And postdoctoral position after that was also on causal learning from unstructured data. And I think, I'm not 100% sure, this was with Yoshua Bengio at Mila, who is otherwise known as one of the fathers of modern AI. And Jason can correct me if I'm wrong. But I think this is the kind of theme on causal representation learning, which he is continuing in his current role in Manchester. And today he's talking about the biological representation learning and connection to the experimental design. And I'll leave it to Jason to explain that. Over to you, Jason. Thank you very much for agreeing to present in our summit series.
Thank you so much for the introduction, and yeah, just repeating, I am very sorry that I'm not there in person. I was planning to be, but both my wife and daughter are down with, I don't know, possibly scarlet fever. We're not certain. So, so there was an outbreak at the school. So, we're yeah. I just couldn't really make the trip up to Manchester. I live in London, so it's especially complicated. Yeah. Anyway, so this talk was originally prepared for a causal representation learning workshop at NeurIPS, or a version of this. It's adapted. Sorry, not NeurIPS. ICML. Gotta get the right conference. And so it was. If it skews to oversimplify biology, I apologize in advance, but I also don't come from a biological background, so you guys can all correct me if I get anything wrong. But yeah, it has scaling in the title, so you would think it would be all modern, but actually it starts 300 years ago when James Lind was hanging out in the English Channel and dealing with the scourge of the day for sailors, which was scurvy. And this was a huge problem for the English Navy, as those who know the history will know. Sailors were left listless and weak and have these big gangrenous sores on them, putrid gums, and in some cases even died from this. So at best it left you with a very weakened group of sailors, and at worst it actually resulted in the sailors not surviving the trip. So there were a lot of home remedies at the time. Captain Cook liked malt vinegar. Elixir vitriol was popular, which is basically watered down sulfuric acid. Sauerkraut was one of the sort of popular home remedies that actually would have worked at the time in some cases, because sauerkraut does actually have some vitamin C in it. Of course, they didn't know that at the time. Cider was popular. Some sailors resorted to eating rats. That was another thing that also actually worked, because rats are able to synthesize their own vitamin C. But of course, these are all just these home remedies. Some things work, some things don't. And another thing that James Lind ended up testing was half a pint of salt water. You drink half a pint of salt water with your meal every day. Seawater. And then of course, lemons or limes, so citrus. And the common theme among these many of these things, aside from the rats, is acids. I think Lind and many of his contemporaries believed that it was kind of like bad food, rotting food was kind of the problem, and so we could eat acids. We could kill the whatever was going wrong, and so you could kind of offset the problem. And they certainly weren't thinking of vitamin C at the time. And Lind famously ran what is sometimes regarded as the first clinical trial, although there's always debates about first of anything. And he had 12 patients, 12 sailors. He split them into groups of 2. each pair got the same thing. So one group got malt vinegar, one group got elixir vitriol, not sulfuric acid, but it's basically the same thing. Cider was one of the things. The salt water was one of the things he tested, and then lemons. And at the end of a week, the only patients who saw any change really was the pair who was having lemons, and they were well enough to help the other patients by the end of a week. Now that was 1747 or 1748. I might be getting the date wrong by a year, but the story didn't end there. So you would think, okay, cool. So we've discovered the cure. In fact, Lind himself didn't even take citrus too seriously, and in part because the kind of effects were kind of unclear. So sort of later, over the next couple of years, he would say, look, citrus seems to help. So they boil up citrus and send it on long voyages, not realizing that by boiling the lemons, you actually kill the vitamin C that's in the lemon. So you're killing the mechanism of action by boiling the lemons. And then the sort of effects were pretty ambiguous. So if rats seemed to help, but if you ate salmon, it didn't help. If you ate tuna, it did. If you ate the captain's pet guinea pig, it wouldn't help. And so there was a sort of like ambiguity over these cures as to what works and what doesn't. So the lemons, if you boil them, didn't seem to work anymore. And it was only sort of the late 1700s that sailors themselves started to realize that they were aware of Lind's trials eventually, and started to realize that citrus just seemed to help, that lemons just seemed to help. But even Lind himself wasn't sort of convinced of this initially. And so it took another 100 years, so this was late 1700s, took to the early 1900s, when Axel Holst and Theodor Froelich discovered that if you fed guinea pigs just a sort of diet of pure grains, they would develop scurvy. And so now we had a model animal for scurvy. So guinea pigs became the first model animal for scurvy. So you could get kind of scurvy symptoms in an animal, which of course lets you now test for potential cures in a more kind of robust, rigorous way. And then it's a pretty similar time, early 1900s. Albert St. Gorgi was studying vitamin C, and was able to feed the guinea pigs a sort of do a proper placebo-controlled trial. Let's say they feed half the guinea pigs some boiled foods, so foods that didn't contain vitamin C, and the other half foods that did contain vitamin C, and show convincingly that was really the vitamin C that was driving the effect or the prevention. Right. So why am I telling you this long-winded introduction? It's really about models, right? So in the 1700s, Lind and those who interpreted with Lind's trials were able to use the results on the sick patients who were involved in his sort of 12-person trial to predict that lemons might help against scurvy. But that didn't give a mechanism. It just gave a prediction, right? So it gives you a sort of prediction of what will happen if you take lemons. And it took another sort of 170 years before Albert St. Gorgi was able to sort of extract the mechanism and show that it wasn't the acids in the lemons that was preventing scurvy. It was really the vitamin C. And once we had that, then Pfizer could go off and manufacture vitamin C at scale and actually put an end to scurvy as a very real problem on ships, right? And so that seems like an ancient history, but it's the case that even today, the overwhelming majority of drugs, over 90% of drugs that are approved on the market, are derived from highly predictive phenotypic models. If you ever want to see a paper that makes you feel lazy as a researcher, this paper that I'm referencing here, there was a single author paper where he went and looked at the approval process for every single approved drug on the market, and looked up the actual story of how the drug came to market, and the vast majority came from phenotypic models. So whether it's animal models or cultures of bacteria, fungi, protozoa, and so forth. So these are models in a physical sort of real world sense. But it's worth talking about them because there is a lot of hype at the moment around virtual cell models. So the attempts to build predictive models that run on a computer of cells, and with that comes questions of what qualifies as a virtual cell. Especially in today's world where we were sort of wanting to rely on machine learning techniques to build these models rather than sort of mechanistic models, which are much older, right? So there's obviously a much older literature from the early 2000s of simulating out differential equation-based descriptions of the cell. But if you're kind of building a machine learning model, which is kind of more black box, when are you done? When have you succeeded? And so my personal take is that I think with any of these desiderata, we'll satisfy them and then realize that much like AI, there are other things we want from a model. So the history of AI was one of saying, if we solve Go, we would have achieved AI, or if we solve chess, we would have achieved AI. And we keep ticking off. If we solve the Turing test, we would have achieved AI. We keep ticking these boxes, and nevertheless have not solved AI in any means. And I think virtual cells will probably be similar, but we can at least describe a couple of desiderata of things that you might want. And this is something that my colleagues at Valence and I wrote down a couple of months ago. So the first sort of requirement is obviously prediction of the response to perturbations, right? So we want models that are able to predict the outcome when we perturb cells in different ways. So whether it's knocking out a gene or applying a drug, and we ideally want this to be sort of modality independent, right? So you can measure cells in many different ways: microscopy, expression data, proteomics. Choose your modality of interest, and you would want a model that is able to predict the response to different perturbations. That's a very hard problem. No one has solved that, but that's at least a sort of something that you would want. Then you would also want, the long introduction around Scurvy was meant to motivate that you'd also want some sort of mechanistic explanation for why we're seeing these predictions, right? And this can be in the form of testable hypotheses that may or may not be true, but at least gives you a way that you can go off and test them. It's not anything. The difference between pure predictions, we can kind of measure the outcome of an experiment and measure whether we were correct. With explanations, with sort of mechanistic explanations, we often need purpose-built experiments, sort of specialist experiments to test those. But you would ideally like some sort of mechanistic explanation for why we're seeing what we want. And then if we have that, then we can move that to we can once we sort of verified those explanations, we get some discoveries which are kind of true insofar as things can be true in science, putting Karl Popper's critiques aside. Okay, very briefly, so that's the sort of what would we want from a virtual cell motivation? My personal motivation for thinking about this just comes from my background in causal inference. We've got sort of the last 300 years of causal inference, starting from sort of the people like Lind and James Snow, who were relying on John Snow, who were relying on experimentation to draw causal effects in the 17 and 1800s. And then, the 70s to today gave us a suite of methods that allowed us to draw causal effects from observational data. And so there's a lot of techniques now that we have from people going about their lives. We can still use that to infer causal effects. But something that all of these textbook methods don't have is the ability to say anything about unstructured data. And actually, please actually interrupt me if with questions. I'm very happy to take questions as we go. So don't feel the need to wait till the end. And so we don't really have good causal methods for unstructured data, like microscopy images, spatial transcriptomics, H&E images. The kinds of data that we generated in bulk in biological applications. we don't really have any kind of good textbook methods that we can rely on to draw causal effects. And so I'm sort of interested in the theoretical question around that. Okay. All right. So with that, long intro out the way, the first part of this talk, we're going to look at how we can kind of represent morphological responses. And we'll start by just saying, what do we want from a representation? Now, causal representation learning as a field has spent the last couple of years trying to sort of say when we can identify latent variables, and so there's been a lot of work on disentanglement. Turns out that's a very hard problem both in theory and in practice, and it's especially hard because you kind of need to commit to a notion of latent variables for your system. Now, I think that many in biology would agree that if we could have a model that could map from an observation of the cell to an abstract latent space that gave us biological features, we'd be very happy. But this is very hard to do because despite the fact that we have a lot of interventional data, even defining what the right kind of latent variables for a cell is in these settings, if I'm measuring a microscopy image, what are the sort of latent variables associated with that microscopy image is kind of unclear. And so what we tend to do at Recursion, where they do a lot of drug discovery from microscopy images, is we have these sort of proxy metrics for, have I got a good representation? The first one is very sort of natural minimum thing you would want, which is perturbation consistency. So what we want is if I do the same thing to the cell in different assays, I'd like to ideally embed that to this, in different measurements of the same assay. I'd like to embed that to the same place, right? So if I've got sort of two images of the cell that are perturbed in the same way, I want my embeddings of that to be cosine similar, the average embeddings for that. And then the second one is I want to recall known biological relationships from the tails of cosine similarity distribution. Okay, so this is a bit more of a mouthful, so let me explain that. If I say knock out two different genes that code for two proteins that form a complex, I expect that if I knock these two genes out, they're going to kind of break the cell in the same way, right? So if I knock out, because essentially I'm just turning off this protein complex by breaking different parts of it. And so ideally, I would like it to be the case that if I look at the cosine similarities between representations for gene I and gene J that form this complex, they should be very similar, whereas, or at least far more similar than they are to any other, to any members that aren't forming a complex, right? And so what we can do is we can say, let's look at cosine similarities, look out the tails, look at the sort of most cosine similar genes, and I want those to recall kind of known protein-protein interactions that occur in biological databases. Okay, does that make sense? I can't really see any nodding heads because most of you don't have cameras on, so but hopefully that's making sense. Okay, so under this metric, it turns out by magic that I still don't feel like we properly understand, that large masked autoencoders are very, very effective at this task, right? Now, what is a masked autoencoder? It's a model that was originally developed for representation learning in sort of natural images. And it's a very simple idea, but very scalable. So what we do is we take an image that looks like this. This is an image of a neuron cell. You break it up into patches. You mask out a fraction of these patches, and it's actually quite a large fraction of these patches. You sort of up to sort of 80, 90% at times. Then you create, you take all these little patches, you feed them through a transformer. You get a representation for each of these patches. You kind of expand out, so you have a representation for the ones that you masked out as well. So you just sort of add add the extra tokens, and then you ask a decoder, which is also a transformer, to reconstruct the original image. And so it's just kind of masked reconstruction, which is an old idea from autoencoders in general, but with this sort of transformer as the encoder and the decoder, and with this kind of patching strategy for masked reconstruction. As is kind of almost cliche to say at this point, if you scale up these settings in very large data sets with very large models, you get better and better representations under these metrics that I defined. So you get more and more recall of known biological relationships. This is with quorum, but this you can look at StringDB as well. Other of this Reactome and StringDB. There were a couple of others that we looked at. So you sort of recall more and more known biological relationships. And then you also get more and more consistent representations. So you sort of represent the cell perturbations in the same way.
Hi, Jason. Before you move on to on the masked autoencoders, I mean, the fraction of masking.
Yes.
How this relates to kind of your learning? I mean, what kind of fractions would work for, let's say, microscopy images? Is it?
You want it very high. So it's surprisingly high. So I think I'd have to, I can't remember the exact number that we ended up on, but it was like 80% plus. And I think when we tested all the way out to 90% of the image mask.
Why is that? I mean, that's my question.
Why?
That? Yeah, I mean, it means you. Is this the reason it learns?
Yeah. So okay. So
if it's low, then does it make it very easy to?
Well, okay, so if it's super low, then it's a very easy task, right? Because in super low, you can kind of learn the identity function. If it's low enough, you're not doing much reconstruction at all. And so the higher it is, the more you need to kind of, okay, with the big caveat that we don't have good theoretical understandings of these representations. So anything I say here is pure speculation. I'm on the record. But if it's super low, you know, you can just basically you can do kind of pixel infilling. Whereas, so this is I'm glad this froze where it did. This is an example of what the masked image looks like. So this is like an actual masking. This is actual reconstruction from layer 0. So you'll see this gets actually surprisingly helpful for this, because what we also found, and this is, I didn't mention this in the slides, is that like intermediate layers, so not the final layer of the transformer, but intermediate layers tend to actually produce the best representations. So you want like about a third in. And what we think is going on here is you kind of, if you mask a lot, you have to kind of have your best guess at what sort of high level structure is going on in the image. And then you have to kind of fill in the detail as you add more layers, right? And so if you watch this GIF as it goes along, so this is like reconstruction from different layers. And so, you know, now we're at layer 13 or 14 of a 40-layer model. And you can see you're just kind of adding more and more detail as we go up the layers, right? And so it's sort of getting the broad stroke structure of the model at the low level layers. So you've masked a lot. And so I need to guess kind of what is the rough structural elements of what's going on in the image. And I have to do a pretty good guess of that because I need to use that to reconstruct. And then sort of as I go further out into more detail, I have to get the fine details right. And so I think sort of there's two parts to the answer. It's the one is like you need to mask a lot in order to force the model to get this like abstract representation of what exists in the image. And then, you know, kind of conditional on that being true, then it has to do the fine details. So which is also probably partly why, you know, intermediate layers help. Okay. Good question. Okay, so this slide is actually, maybe I should have showed a bit earlier to sort of explain these cosine similarities a little bit more. So it's quite a nice example of how kind of nicely this kind of thing works. This is actually with an older model, so but these newer models do better on this metric, so you'll see similar results. So if you look at something, you look at a big complex protein complex like the integrator complex, which is made up of a number of proteins, and you look at like these, and each protein kind of forms different modules within this complex. You can then look at the corresponding embeddings in these representations. It's quite amazing how you can kind of see, you can even see the individual modules in the embedding. So for example, if I look at this enhancer module, which is made up of these three genes, anti-S 13, 14, and 10, and then you look over here, there's the anti-S 13, 14, and 10. And you can see these are highly cosine similar to each other. They're less cosine similar to the other modules, but still more than than other genes, and this would still, these would still be redder than your than a randomly chosen gene in the matrix. And so you're getting kind of both kind of within complex structure in that there's the sort of enhancer module and you're seeing similarity, and then you're getting that the sort of enhancer module is more similar to what's it, like INT7, where's that? The central backbone, and indeed these two things are neighbors within the protein complex. Now, this is a particularly good example. This is sort of taken from Recursion's corporate deck, so it's cherry picked, but I do think it's still pretty amazing that you're getting this from microscopy images alone. And I'm going to pause for 1 second and grab a charger because my laptop is going to die while I'm speaking. So take a glass of water. I'm sorry. I thought I had more battery than I did. All right, that was embarrassing, but good. We are going to survive the talk without a dead computer. Okay. Why do I show you the slide other than that the fact it's cool? I sort of said we want mechanistic explanations, and I think this is a nice example of this because this particular gene wasn't characterized at the time the study was done, but in later studies, it showed that there is actually another protein that forms part of this complex, and this gene codes for that protein. Recursion wasn't involved in this; it was sort of done independently, but it sort of suggests that it is possible to use these assays in order to to find other potential protein-protein interactions, and I think that's a kind of cool direction. Okay, so that was like learning the representations themselves, but it reduced everything to these cosine similarities. What if we want to sort of break apart these representations into sort of give a more fine-grained description of this, and so if two things are very similar, cosine similarity will just tell you these things are very similar, but when they differ, you kind of don't get anything other than the fact that they're not cosine similar. You don't get any notion of like how they differ, and you don't get a sort of any kind of disentangled representation. And so kind of in parallel to the work that's been done in causal representation learning, thinking about disentanglement, there's been this other field of mechanistic interpretability that has sort of made this, that has started from the assumption that the way neural networks solve their problems is by decomposing representations into features that correspond to directions, right? And so the sort of starting hypothesis, which may or may not be true, but we just take it on faith in mechanistic interpretability, is that you have some representation space where you have many different meaningful kind of feature directions, and that's just what the neural network has learned, right? And so most of the prior work in this area has done this on language models. So So you use sparse autoencoders or dictionary learning more generally to decompose these representations into into like a sparse set of features. And the reason for this is the sort of central hypothesis is there are many more features, so many more directions than there are kind of dimensions in the hidden layer of a neural network. So, for example, a language model might have 4,000 dimensions in its hidden representation, but it can represent, millions or billions of different concepts, right? So if the way that representation worked was like, well, I have one concept for man, one concept for woman, one concept for dog, one concept for cat, I'd very quickly run out of concepts, right? If it's, if I've only got 4,000 dimensions, I'd be kind of stuck. But if the way these models are working is actually, it has the sort of sparse set of features, well, the set of features, concepts, which is maybe billions of concepts, but only a few of them are on at any one time. Then you can take results from compressed sensing to say, look, if I've got this very large vector that is sparse, I can compress it into a lower dimension. So go from like 1 billion dimensions down to 4,000 dimensions. And then I can recover those sparse directions using dictionary learning. Okay. So that's the sort of basic hypothesis. And it turns out in language models, this works surprisingly well. And so we were curious whether the same thing applies in these big microscopy foundation models. So this is work led by my fantastic intern, Konstantin. And Christina was also very involved in helping supervise it. So she did all the cool biology work, and he did all the cool dictionary learning work, and so they were a great team. So by default, they use a lot of these methods use sparse autoencoders. Turns out those didn't work so well for the microscopy images, or they didn't work as well as what we came up with, which was this matching pursuit-based algorithm, which I'll explain in the next slide. Actually, in the interest of time, I might skip over that. But essentially, you can kind of decompose these features into different directions, and you can learn meaningful things. Okay, I'm going to go over this quickly because I realize I'm going slower than I should, given how much I want to cover in this talk. Okay, so the basic idea is I have some dense representation, which is just, you can just think of some embedding of the image, right? So this is like the final hidden layer or some hidden layer of the transformer that gives a direction in embedding space. And I want to decompose this into meaningful features. Now, as an inductive step, just assume I have these features, and we'll come back to where they come from. So if I have a set of directions, so features correspond to directions. In matching pursuit, what I'm going to do is I'm going to find the direction, in this case D3, that is most similar to the feature direction of interest. And then I'm going to regress these. So I'm just going to scale this out so that I minimize this error over here. And then I'm going to look at the residual. So x1 becomes the residual of the sort of regressed feature, right? And then I'm going to iterate this procedure, right? So I'm going to find the sort of direction that is most similar. So that'll be this one. Regress it, so scale it out, and then I'm going to find another direction and so forth. So I'm going to sort of iteratively add features of that form. It turns out this is a procedure that you can just iteratively run, and then you can use gradient descent to learn these feature directions. Right, and I won't go into the details, but it's just, you can literally just do gradient descent on this dictionary. There's one more trick that we have to do, which is that in normal cellular function, there's a lot of like biological variation that you're not that interested in. You're interested in the differences across perturbations, right? So cells go through cell cycle. There's just like the sort of layout on the plate. There's also sort of normal variation, and so you want to kind of squash down the directions of variation and accentuate differences across perturbations. And the way we do that is by applying PCA whitening, where you compute the whitening kernel on the control cells, so cells that weren't perturbed. And so that's going to kind of squash down normal variation that happens to control cells, and it's going to and then when you apply that whitening kernel to the other cells, to the perturbed cells, that's going to sort of expand out the variation along those dimensions. Ask me questions if you'd like more detail on that, because I know I went over that quickly, but that's essentially the idea. Okay, so once you've done that, what do you get out? Well, you get out sort of, at the minimum, you get sort of some fun, non-biological features like light leak feature. So when the microscope has the light leak, we have a feature direction associated with that. We have a dead cell feature. So when we have very empty plates, in fact, this was the very first one we looked at. We thought the method was broken because we kept coming up with these like blank images, but it turned out it was just like perturbations that killed a lot of cells. Then multinucleation features, which is a little bit more biological. So there's like a feature direction associated with images of cells where there's more than one nucleus in the cell. Some of these sort of interesting cell structure type features that come out. And then this one's just kind of fun where Christina went in and looked in detail at one particular feature. So, okay, we can take a gene of interest. So in this case, OPA1, and we can look at the average embedding of its sort of feature vector. So I know I've got a whole bunch of images from knocking out that gene. I compute the embedding associated with that gene. And then I can say which of my learned dictionary feature directions is most similar to this gene, right? And so in this case, it would be DI. And then because it's a masked autoencoder, I can look at each little 8x8 patch because the masked autoencoder breaks the image up into little patches. And I've got a representation for each of these patches, and I can see which of these is most aligned with this gene's feature direction. And then I can ask, I can see that, some of these are very not aligned. So these ones are kind of orthogonal to it. This one's kind of more aligned. And these guys are the ones that are most aligned. And if looking at the image, it's kind of not obvious at all what these are pointing to. But if you look at the mitochondria channel, so these are cell paint images where you sort of have different channels, you can see that sort of the ones that are most aligned are where the mitochondria are lighting up a lot. And then you can go and ask your favorite biologist, your biologist friend, or ChatGPT if you're like me, and ask, what OPA1 does in the cell, and it's a gene that's responsible for mitochondrial function. So again, this is a sort of specific example where we've looked at a gene where we know the function, we're checking the representation is working for that function. How consistent this is across all genes is still to be to be determined. But there are at least examples where there's a very nice correspondence between the features we learn and the biological function of the cell. So yeah, I sort of said this. I said this. Like I think these dictionary learning kind of approaches are really interesting. They give you sort of some meaningful features, and there certainly are some that correspond to biology, but it's kind of very unclear how many and like how much your coverage is like. And this is a problem of dictionary learning generally. This is true in language models and it's true in biology. So it gives you sort of a set of interesting directions, but it's not clear where you get all interesting directions or all biology. So there's still a lot more work to be done on this. Okay. With 20 minutes left, let's switch gears now to perturbation effect prediction. Actually, before I switch gears, I'm going to pause to see if there are any questions. All right. No. Okay. All right. So that first part was on explanations or sort of mechanistic hypotheses that we can debate about. But I think, if we want virtual cells, the sort of minimum that I think everyone agrees on is they should be able to handle predicting the effects of perturbations, right? And that is very desirable, but there's been a bunch of recent papers. This one has a particularly nice figure that show that these models are actually pretty bad at that. The current generation of foundation models. So there was this paper from Konstantin Alman-Elter, who showed that if you just use a simple additive model, which I'll explain in a bit, that outperforms, SCGPT, SC Foundation, Gene Former, GEAR, CPA, many of these methods that have gotten a lot of attention in predicting the effect of a double perturbation. Right, and so this is specifically in double perturbations where this is most bad, although there have been sort of analogous results in single perturbations as well around, the competitiveness of just mean baselines. My take on this is, in double perturbations in particular, I think we've got a kind of wishful thinking if we think this is going to work well. These methods, this particular slide was made on this Norman et al. Data set, where there are only 124 double perturbations, of which they used, I think it was 60 in order to do training, and the other 60-ish to do validation. We're training models that have 1,000 dimensions, and so in the sort of double perturbation setting, we're really in the sort of big D, small n regime, which has never worked well for neural networks. It's not. So I think this. We really do need to think about how do we expand the space of double perturbation data if we're going to do well on this task. That said, the reason we don't have a lot of double perturbation data is the space of double perturbations is vast, and so we need to think about how do we choose which experiments to run. So to put numbers on this. There are around 20,000 genes in protein coding genes in the human genome. At a place, it's increasingly common to get whole genome knockout screens. So in my recursion life, they do this regularly, where you can screen all 20,000 genes on the human genome. But if you want all pairs of genes, that's on the order of 200 million experiments, and that's way larger than any lab can do at the moment, right? That's sort of many orders of magnitude. Bigger than the largest experiments that are currently being run. So we can't run all pairs of experiments. That's just kind of a hopeless task, and so we need to be smarter about how to choose this.
So sorry to interrupt, but this kind of if I say entanglement is pretty. It's not going to stop at 2. I mean, this is the entanglement actually is multidimensional, right? So.
You mean, well, okay, so I mean, this is like a lower bound on the kind of a very, very pessimistic, very I guess optimistic lower bound on the space of experiments. If you go to compounds, then you're in, then you're into even if you just take a library of 50,000 compounds, you're into 1 billion experiments. If you're going all compounds versus all genes,
so need for your method will be even more. What are you going to do?
Yeah. So I mean, it just gets kind of bigger and bigger and bigger. I'm sort of being as generous as I can to what brute force would look like, and even there, it's infeasible. But if you into the space of all possible synthesizable molecules, then you're just in billions and billions and billions. It goes, and then you can also multiply by the different kinds of assays you have and so forth. So there's just a lot. Okay, now. I think it's worth thinking about. So if we want to think smartly about how to design experiments, this just takes us into the realm of efficient experimental design. But we're now in this new era where we're collecting this kind of unstructured, messy data and using these big, ugly foundation models. And so it's worth contrasting these two, the way we approach these two problems. So in traditional experimental design or active learning or Bayesian optimization, it tends to be task specific. So your sort of starting point have some specific task that you have in mind, either implicitly or explicitly, because you need to define a utility function, right? And that's sort of the starting point of any of these methods. It tends to be model specific, because once you've defined your utility function, you say, what is the optimal experiments that would help be most informative with respect to the parameters of my model for this task. And then often in practice, there's kind of a lot of modeling constraints, because you want to make these information gain computations tractable. And so you end up constraining the class of statistical models that you're allowed to use for these tasks. By contrast, with foundation models, we kind of expect generality. We expect task agnosticness, right? So whether we actually achieve that is a whole nother question. But at least in principle, what the claim is that you train a foundation model and it can be used for all tasks and not just for one particular task. So we sort of want generality by default. This space is evolving quickly. There's kind of constant updates to these models. And so ideally, you would like the data that you collect to be useful for all models and not just for some specific model that you're training now. You want a bit of future proofing in the data collection process. And then because these models have billions of parameters and highly nonlinear and so forth, any hope of kind of tractability and closed form solutions, I think, is out of the window, right? So we kind of want, in this era, we want like an experimental design technique that doesn't know what the data is going to be used for, that doesn't know which model is going to be trained on this data, and is computationally efficient. And so, it's kind of like we've thrown away all the structure of the problem that makes experimental design useful. And, or at least it appears that way. And so what I'm going to argue in this is that there's still kind of hope. And this is work that has kind of been going on for a while. The first version of this paper has been on archive since 2024, led by David, who's fantastic, and Mox, who's also fantastic. And Scott, who's done a lot of the most more recent experiments, and then a lot of other colleagues. Okay, now I come from causal inference. We like hierarchies in causal inference. And so I'm going to give you a hierarchy of tasks that you might want to solve, and then argue how you can go about solving them. Okay, and I'm not going to tell you yet how to solve them, but I'm just going to say this is a kind of set of things that we could try and solve in this world. So the most general thing you could ask for is that I can predict the double perturbation distribution, P of X given ij. So if I and j are two different perturbations, I can just characterize that distribution. Because if I can characterize that distribution, then I can answer any query about that distribution. So any task on top of this distribution is just some function that I apply to this distribution. Right? So kind of if I solve this, I'm done. Then the next step down is I say, look, I know that the form of the query that I'm going to be asking is in the form of a mean embedding, right? So what's a mean embedding? It's just the average across this distribution of some function h, right? So sort of any query that I ask is going to take the form, well, I have some function in mind, I'm going to average across all the data. So like if I had run the experiment, I'm going to apply some like embedding function to that, and I'm going to average it. And then I'm going to just do something with that rather than doing something with the distribution of a whole. Now, it turns out that's just a more constrained setting, and we can approach it differently. So it's more constrained, but it's sort of useful for many things. But importantly here, I'm not assuming that I know h, right? So I could do this for sort of any embedding function, not some particular embedding function, which then suggests the kind of next step down on the rung is where I know h as well. So I know the embedding function I'm going to be using, and so that's a more specific case. And then finally, the most specific, sort of most constrained case on the hierarchy is where I just have some scalar property of interest, right? So classically, this would be cell count. This is something that's often studied. I'm interested in the effect of a perturbation. What does it do to cell viability, right? And so, cell count you can think of as some very specific function, which is like take the measurement that I ran, count up how many cells, that's my function. And then if I'm interested in like average cell count, then that would be like a very specific choice of h, right? And puts us back in like a land that we know how to solve. Okay. And the way we're going to approach this is that at each rung on this task hierarchy, we'll say that if perturbations don't interact by some notion of interaction, which we're going to define in a minute, then we can solve the associated task from single perturbation experiments, right? So we're going to sort of say, look, if things don't interact, then I only need to run the single perturbation experiments, and I can predict what's going to happen in the double perturbation experiments. But if they do interact, then I have to run the double perturbation experiments. So we're going to kind of break it up into these two cases. And so to kind of convince you that this is, maybe has some familiarity, we'll start with the simplest case on the, the rung one task, which is, do drug combinations have a synergistic or antagonistic relationship, right? So classically, this is studied in oncology. You're asking whether two cancer drugs have a synergistic or antagonistic effect on the cancer cells. And we define these things to be bliss independent if the survival probability of applying both drugs equals the product of the survival probability of applying drug I and then drug J, right? And so this is just a definition of independence. It's subtle, but if you ask me, I can explain why. You also kind of need the control. Like if some cells are going to die automatically, like on their own without you in the control world, then you have to account for that. You don't normally think about that because cancer cells are normally growing, not dying, but you sort of you do have to account for that to be formally correct. We can equivalently write that as in this form, and the only reason for that is to kind of draw an analogy with what we're going to do later. Okay, so we can equivalently write that in this as a sort of log Pij equals the sum of log Pi plus log Pj minus log P0. Okay, so that's like a definition of independence, and it's sort of definition of non-interaction, and this is done very widely in just discovering whether you have synergistic or antagonistic effects. Next rung up on the hierarchy is where I have some specific embedding function. In the simplest case, this is just the identity. So if I'm working with single cell expression data, an example of an embedding function would be pseudo bulk, which is where I just like take the average number of average expression or the sum of the counts. And then in this case, non-interaction that we can define is when the so-called additive model holds. Now, what is the additive model? That's what we saw worked so well in Constantine's paper, which is where you take the single perturbation, subtract off the control embedding, and then to define a vector direction. So this is like the direction associated with gene I, direction associated with gene J, and we just add these two vectors, and you get the direction associated with gene IJ, right? And so here we're defining non-interaction as being when when the additive model is going to work, right? So that's just another notion of non-interaction. And this can be defined, it can be defined for any embedding function. Okay, now I haven't told you about rank 3 and 4, we'll get there, but I'm just going to first explain sort of what we're going to do with this definition. So I said we can kind of separate things into two cases. One is where we know we assume that things will work if things don't interact. So we have a notion of non-interaction, and things will work insofar as they don't interact. And then we're going to derive a test statistic that tells us whether or not things will interact. And I've got the 5 minutes left notification, so I've got to hurry up and finish off. Okay, so and then we'll collect some data at various points. So we can collect, some fraction. So you can think of this kind of matrix of all perturbations, all IJ perturbations. If I collect a sort of set of IJ pairs, I can measure the test statistic in the places that I've collected. So I can measure a test statistic saying, do things interact or don't they? And then I can estimate this test statistic for all unseen pairs, and I can select the unseen pairs that are most likely to interact or have the largest interaction score. Okay, and in doing so, I'm going to be collecting data which is very unpredictable from the single perturbation experiments, and I can rely on the single perturbations to predict where things are, where I'm going to generalize. Okay. Now, with 5 minutes left, I'm going to try and explain these other two concepts. So we have these two notions, disjointness and separability. These correspond to the sort of mean embedding case and characterizing the distribution directly. So we'll start with disjointness, which is when can we characterize mean embeddings. So as I said, a very common practice for in this case, which is this additive model was an example of this, is to represent a double perturbation as the sum of single perturbations, right? So I'm going to represent this sort of IJ embedding as the sum of this averaged perturbation I and the average of perturbation J, always centered by the control average. It turns out if you just do the algebra, you get this relationship over here. Okay. So you get kind of this word vector style analogy. So this like man is to woman, king is to queen kind of thing that is common in NLP as well. Now, a sufficient condition for this to hold for any embedding function H. Oops, sorry. The sufficient condition for this to hold for any embedding function H is if you have the densities, these conditional densities summing. Right now, this is a very weird condition. We can talk about it some more. I can explain some cases where this happens. But if this holds, then by just taking expectations, it's easy to see that this is going to hold. Okay, so sort of if I have this condition happening, then for any embedding function h, we'll have this algebraic relationship, right? And so we can turn this into a statistical test by sort of multiplying that relationship across by 0.5. So now I have one mixture distribution that I have to compare to another mixture distribution. And I can use MMD test, which comes from Arthur Gretton and his co-authors. And so it's just a test of where the two distributions are equal to each other, right? And so I can create one distribution, create another distribution, and I'm testing whether we have equality, right? So that's just a sort of very simple null hypothesis, and then we can run MMD to test whether this is the case. So one thing that's most important to emphasize here is we're testing a property of the data. So this is like purely a property of what's being generated and not a property of any model that we've chosen, right? And so this is going to hold for any particular architecture that you apply on top of this. So that was the disjointness. Separability is this other condition, which is sort of very analogous. So we want to, but here we want to characterize the whole double perturbation distribution, right? So this is so rather than just working with mean embeddings, we're just going to characterize the distribution. And it turns out that the sufficient condition for this is if the log densities sum. So this is very similar to the condition I showed you before, except we have log densities instead of densities now. Sorry, I just realized my clock is showing me I'm very much out of time. I'm sorry. My clock on my computer is showing me quarter to 3, not 3 o'clock. So I looked at my watch. I'm going to just say you can do this, and let me conclude here. That's really embarrassing. Sorry, my clock is showing the wrong time. So I thought I had 15 minutes.
I'll just mention that I know. I mean, since we are fully online, we can stay a few minutes more if somebody has any questions.